everyone, welcome to Aakash. This is Lavina Singh, your botany faculty, and resuming the session with slot 2 for AIATS Plus Phase 2 Mock Test 8, which was conducted on 9th of March 2025. I hope you guys are taking good care of yourselves. आपकी अच्छी तैयारी हो रही होगी आपके NEET exam के लिए. It is just overhead only, and इस time पे you people are polishing yourselves, also preparing for your board exam, and a lot of pressure. I know this month is like that only, but we need not to build pressure. We need to keep calm and stay focused, right? And over here we have our next slot, that is slot 2, in which I will be taking up the questions in which you make frequent mistakes, and the chapters are biological classification, anatomy of flowering plants, plant growth and development, molecular basis of inheritance, as well as biodiversity and conservation. All right. A very good afternoon to you, Harsh. So, in this session, as you people already know, this has been a long series now, and we discussed the questions in which you make frequent errors, right, so that you do not repeat those mistakes, right. We discussed the question as well as the related concepts with you, okay. So, in this session, let us begin with the very first question, and the question is question number 92 from your test paper, okay. So, it says, which of the given character is shown by all members of Protozoa? So basically we have to focus on protozoans, right? Right. So to which particular category these protozoans belong, which kingdom they fall in, right? We have to see that. I will discuss the characteristics with you first, okay, and then we will come back to this question, okay. Very good afternoon to you, Aman. Now, we know that while we are speaking of protozoans, they are belonging to the kingdom Protista, okay. So, if we are taking up kingdom Protista, we basically divide it in 3 categories, okay. So, the 3 categories that we speak of are, first of all, the photosynthetic protists, okay. That means we are talking about the organisms that can prepare their own food, okay. They are autotrophic in nutrition, okay. So, we have photosynthetic protists. Okay, the next category that we take up is the ones that are consumers, okay. That means they cannot prepare their own food, as well as they are parasitic, okay. So, this is the second category. And the third one belongs to protozoans, right, from which we have been asked the particular question. Now, you'll say that why protozoans are not put into the category of consumers? After all, they are also consumers. They cannot prepare their own food, but they are not parasitic, okay. So, we are putting them in the third separate category, okay. Very good afternoon, Devil Chaudhary boy. Important topics for boards. See, genetics is very important. You must be given the whole distribution from CBSE, okay. So, the weightage of different units is different, okay, and there are so many important topics that are out there. I cannot discuss all of them elaborately over here, but yeah, do prepare your genetics well, do prepare your ecology well, okay, and up to वैसे भी the number of chapters are less, okay. So, you should prepare properly for each and every topic. It is, and it has nothing to do with that this particular thing is important for boards, okay. The pattern keeps on changing, the questions keep on changing, okay. So, you have to prepare pretty much everything, okay. And we can have a session about this very thing, a live session in which we can discuss all the important topics with you, okay, so that you have a better clarity. But now exam is overhead, I will suggest that please prepare your exam from the NCERT thoroughly, okay. Or you can take the help from the previous year questions. You can see which are the frequently asked questions from which very units, okay. All right, very good afternoon, Snehal. Coming back to the topic, we have protozoans, right. Now, if we take up the examples, okay, protozoans have not been put in this particular category because they are not parasitic. They are just heterotrophic, okay. Now, we have photosynthetic protists, there are a few examples, or we can say that if we consider diatoms, okay, so diatoms are there, right, as well as we have dinoflagellates, okay. These are the two examples that we can take for this particular category. Then comes consumers. We have slime molds. Okay, slime molds are basically parasitic, right? And if we are talking about protozoans, right? Now these protozoans are basically divided into 3 subcategories further. Okay, so here we have ciliated protozoans, we have flagellated protozoans, okay, we have the next category that are okay, we have amoeboid ones, right, and then the 4th category, that is sporozoans, okay. Now these are the different groups in which they have been divided. Now if we see the question again, what the question was asking, given character that is shown in all the member of protozoans, right? Now of protozoan, we know the kingdom is Protista, okay, and the very peculiar thing about kingdom Protista is all the organisms, if we are taking up the kingdom Protista, all the organisms are unicellular eukaryotes, okay, they are unicellular eukaryotes. Okay, so from this very thing, it is clear that the option is first. The correct answer is first. But we will go through the rest of the choices also. Autotrophic nutrition. As I told you, the nutrition varies, right? Nutrition varies. They can be photosynthetic, they can be heterotrophic, they can be parasitic, okay? They can be saprophytic, okay? So, the autotrophic nutrition is not something that is applicable to all of the protozoans. So, this is not true, okay? And because they are asking about the protozoans, they are basically heterotrophic, okay? Over here, they are specially heterotrophs. They cannot prepare their own food, okay? They can show holozoic mode of nutrition. They can be parasitic also, okay? Then comes spore formation. Now the category that is sporozoans. Only sporozoans are the protozoans that can produce spores, okay? That are involved in spore formation. So definitely this is also not true because it is not applicable to all of the protozoans. And the last one is silica shells. Now if we are talking about the amoeboids, okay, there are some amoeboids that are found in the marine environment. They have this special feature that they have the silica shells, okay. So if we speak of these protozoans, so it is only applicable to the amoeboid ones that are having silica shell, okay. So ciliated, flagellated sporozoans, they are not having the silica shell, okay. So that is why this is also not true. So these 3 can be simply ruled out, and unicellular is something that is applicable for all of the protozoans that are falling in the kingdom Protista, that is of unicellular eukaryotes. Okay, clear with everyone? Now moving on to the next question. Question number 99 says, read the following statements. The first statement is, monocotyledonous roots have fewer xylem bundles. Monocotyledonous roots do not show secondary growth. Dicot root has small pith. Cortex of stem consists of thick-walled parenchyma cells. Okay, so most of the statements are from roots. Okay, so I am going to discuss the structure of root, the anatomy of root in brief with you, so that we can revise this particular topic. Okay, so let us see that what do we understand when we are differentiating the anatomy of the dicot root or the monocot root. Okay, so I will take up the characteristics of dicot root, and along with that I will be comparing them with the monocot. Okay, so the very first we will begin from the outer side, okay, from the epiblema. So the outermost layer, that is epiblema. In case of root, we call it epiblema. In case of stem, we are calling it as epidermis, or leaf, we call it as epidermis. But when it comes to root, we are using this special term, that is epiblema. Alright, so we have epiblema. Basically, this is, what are the characteristic features? It is the outermost layer, obviously. It is forming the covering, okay. It is bearing root hair. Okay, when it comes to the epidermal tissue system of stems, okay, we find the presence of the stomata, right, presence of cuticle. But in this case, in case of roots, stomata and cuticle, they are basically absent, okay. They both are not found in case of roots. Now this is the epiblema, that is the outermost layer. It is similar for both monocot as well as dicot. Okay. Moving on to the next, that is epiblema के बाद जो हमारी next layer आएगी, that is of the cortex. Okay. Now cortex is basically, first of all, it is parenchymatous. Okay. The cortex is parenchymatous. In case of stems, we study about hypodermis also, but in case of roots, there is no hypodermis, okay. So, cortex, and it is parenchymatous, and the innermost layer of cortex, it is called as endodermis, okay. This is the endodermis. Both in case of dicot or monocot root, the endodermis is basically single layered. Okay. Secondly, it is made up of barrel-shaped cells. Okay. This is a very important characteristic feature. Okay. So, the cells are barrel-shaped. I already have discussed in the previous lectures also this particular structure with you. Barrel-shaped cells. And these cells are basically having the deposition of suberin, okay, which makes these cells impervious to water. This is a characteristic feature for endodermis. We all study, okay. You have to remember these layers properly. एक sequence में आप इन्हें याद रखिए. It will be beneficial for you, and the characteristics along with that. And endodermis का जो important characteristic है, which is often asked, is the presence of suberin, okay. So these cells are basically suberized, okay, and this suberin deposition is forming the Casparian strips, okay. I am discussing this in brief with you. We have already discussed it previously, that there is the presence of suberin, okay, and that leads to the presence of Casparian strips. So, Casparian strips is a characteristic feature of endodermis, okay. Casparian strips are present, okay. So, it is up to endodermis. Now, you can see that it is pretty much same for dicot and monocot, right. Now, if we move to the next layer, that is, okay, after endodermis, inner side, towards the inner side of endodermis, there is the presence of the next layer, that is of pericycle, okay. Now, pericycle, if we consider the pericycle of the dicot root, okay, this is having a very important function. First of all, it is parenchymatous. Again, it is made up of parenchymatous cells. We are speaking about roots, okay. So, there will be the layers that are mostly parenchymatous in origin only, okay. So, these are parenchymatous, the cells are parenchymatous. Secondly, it can be single to multiple layered. It can be single to multiple layered, okay. Now, what is the function of this particular pericycle, okay. The function is basically, it helps in the growth of lateral roots. Okay. This particular function is common for both dicot as well as monocot. Okay, but the next function that I'm about to tell you is it initiates the formation of vascular cambium. Okay, this is very important. And this particular thing happens during secondary growth. Okay, so this is the secondary growth feature, and we know in case of monocots there is no secondary growth, right? So will there be the formation of vascular cambium with the help of the pericycle layer? No, because secondary growth is not occurring in case of monocot roots. So this is only applicable for dicot. Okay, now comes the next layer, and the next layer after pericycle we are considering the conjunctive tissue. Right, now if we are taking up conjunctive tissue, first of all you should know what is the location of conjunctive tissue. Okay, in this these particular representations, you can clearly see there is xylem, phloem, because they are arranged radially. So in the see, there is one xylem and the other xylem. Between two xylem, right, there is the presence of phloem. So between xylem and phloem there is the presence of conjunctive tissue. Okay, so present between vascular bundles. Okay, and if we are speaking of dicot root again, it will help in the secondary growth. Okay, so this you should remember. Next we have the vascular bundles. Now here we draw the difference. Vascular bundles, while we are taking up dicot root, in case of dicot root, vascular bundles are, first of all, they are exarch. Okay, that means the protoxylem will be towards the outer side, will be towards the periphery. Okay, that is the exarch condition. जो छोटा वाला xylem है, okay, that is towards the periphery, and जो मोटा वाला xylem है, that is towards the center, towards the pith. This arrangement is the exarch arrangement. Okay, so exarch condition is true for both dicot and monocot. You can simply see in the representation properly. Okay, but if we are taking up the next one, that is the number of vascular bundles. Okay, so in case of dicot root, the number of vascular bundles is 2 to 4. Okay, they can be 6 also, but generally they are 2 to 4. But in case of monocots, we see the number of vascular bundles is generally more than 6. Okay, so it shows polyarch condition. Okay, there are many vascular bundles, that is more than 6. You can see over here the red patches that you are observing over here, there are more in number. Okay, so basically vascular bundles, we can draw one difference. From pericycle we can draw another difference between these two, and the last one is the pith. Okay, I have little space over here. I am writing about pith over here. In case of dicot root, the pith is very small. Okay, and it is inconspicuous, sometimes not even visible under microscope. Okay, so it is small and inconspicuous. But if we are considering the monocot root, the pith is well developed. It is large and well developed. Okay, so these are the some differences that we have between the dicot root and the monocot root. I have to summarize all of it in front of you. I know it is not entirely related to the question that has been asked, but we are discussing the related concepts. So I discussed the brief of dicot and monocot root. Now coming back to the question, the first statement says monocotyledonous roots have fewer xylem bundles. Okay, over here we are simply seeking that the difference is true or not. Okay, so do monocot roots have fewer xylem bundles? No, they are having more xylem bundles. Okay, so the first statement is not true. The first statement is incorrect because monocot roots have more number of xylem bundles, more than 6, okay, as compared to the dicot roots. Secondly, monocot roots do not show secondary growth. That is absolutely true. There is no secondary growth seen in case of monocot roots. Third statement says dicot root has small pith. Yes, that is true. Abhi मैंने आपको बताया the pith is very small and inconspicuous in case of dicot root. So this is true. And the fourth statement says cortex of stem. Okay, over here the stem has been given. Cortex of stem consists of thick-walled parenchymatous cells. No. While we are speaking about the structure of cortex of a stem, the cells are thin-walled and parenchymatous. Okay, so the folly lies here. It should be thin. Sometimes we read the statements quickly, okay, and we do not pay attention to these minute points, okay. So please pay attention, please read the statements very carefully when they are asked in your exam, okay. Now we have to choose the correct statements, okay. Also pay attention, they are asking about correct statements from the above, okay. So the correct statements are B, C, and okay, we have the correct answer, that is option 3, B and C, because A and D are not correct. Okay, I hope this is clear with everyone, and if there is any doubt, you can simply drop them in the comment box. Okay, moving on to the next question. We have, okay, one of the first gibberellins to be discovered and remains the most intensively studied. Okay, so yes, it is a very simple question, I know, but still you people have made a very frequent error in this. Okay, so the answer is obviously GA3, that is a clear-cut answer, but I would like to discuss the related concept with you, how it was discovered, who discovered it, and few roles that are played by gibberellins in plants. Okay, so let us take up gibberellins quickly. Gibberellin is a plant growth promoter, first of all. Okay, we know while we are talking about the plant growth regulators, okay, that means the PGRs. Among them, see, growth can be regulated in both the ways. It can be inhibited also, right, and it can be promoted also. So there are certain hormones that are inhibiting the growth, like abscisic acid. There are certain hormones that are obviously always promoting the growth, right, such as cytokinin, such as gibberellin. Okay, so if we are talking about the plant growth regulators and we are considering our gibberellin, so it is a plant growth promoter. Okay, they fall under the promoters category. Now, if we are taking up its discovery, basically how it was discovered, it was first of all, the disease. There was a disease that was caused by a particular fungus. You know it. Tell me in the comment box which one is that. Now, that particular disease, the effect of gibberellins was first observed in Japan. Okay, in Japan, the farmers observed that some of the rice seedlings, they grew excessively tall. Okay, so we are talking about the rice seedlings. Okay, and these seedlings, what was the defect? The defect was they grew excessively tall, they were weak and also sterile. Okay, so such kind of seedlings were growing in the fields in Japan. Okay, and they called them, because of their excessively tall nature, they called them as foolish seedlings. Okay, foolish seedlings. Obviously in Japanese they won't be calling it foolish. We are deriving the meaning of it. That meant foolish seedlings. Okay, and the disease was named as Bakane disease. Right now, scientists were very eager. They wanted to know that what exactly is leading to the particular rice seedlings to become foolish, right? What is leading to this particular disease? Then came E. Kurosawa. Okay, what E. Kurosawa observed that this particular disease is being caused due to an infection that is because of a particular fungus that is Gibberella fujikuri. Okay, and yes, the name of this particular plant growth hormone is in the name of this particular fungus, Gibberella fujikuri. ये question भी आपसे पूछा जाता है. Okay. Okay, since it is our scientific name, we need to underline it separately. Gibberella fujikuri was the fungus due to which this particular disease is caused. Okay, later on the extracts from these fungus were taken and they were used for the promotion of the growth of the plants. Okay, they were used in the beneficial activity. Okay, and since then there are hundreds of gibberellins that have been discovered. Okay, so there are more than 100 gibberellic acids that have been discovered. Out of those, the most extensively studied one is GA3. Okay, they are named as GA, 1 gibberellic acid, 1 gibberellic acid, 2, 3 and so on. Out of those, gibberellic acid 3 is the most studied one and one of the first one to be discovered. Okay, now what are the functions? Because we are doing this particular discussion about gibberellins, I also need to discuss few important functions that are often asked in your NEET exam. Okay, so what are the important functions that are performed by gibberellins? Okay, the first and frequently asked one is they basically promote the process of, they basically lead to the bolting process. Okay, so they are useful in bolting process, and in which particular plants this bolting is taking place? In the plants with rosette habit. Okay, I explained the concept of bolting in the previous class. If you have any confusion, go and check it out. Okay, second, it hastens the malting process. Okay, again malting process I have discussed with you. It is done in the brewery industry. Okay, for that gibberellins are used to hasten this particular process. Second, third one is they help to basically stem elongation. Okay, right, they are very beneficial in stem elongation, basically in the internode elongation, and that leads to the high yield of sugarcane also, because the yield that we derive from sugarcane is from its stem, and if we are enhancing its length, so obviously the yield will be more. Okay, so this is another function, and then it is antagonistic to abscisic acid for seed germination. Okay, so seed germination is basically hindered or stopped by abscisic acid, but this particular hormone gibberellin, it promotes seed germination. It promotes seed germination. These are some of the important functions. There are several other functions that are being performed by gibberellins, but these are the features or the functions that are often asked in your NEET exam. Okay, the process of bolting, malting, stem elongation, okay, and promote seed germination. It is antagonistic to abscisic acid because of this very particular feature. Okay, so these are certain features that you have to keep in mind. All right, and there is one more, that is it enhances the shelf life. Okay, it basically delays the fruit ripening process. Okay, so if the fruit is ripened late, it will reach the market late. Okay, and hence it can be distributed throughout the season. Okay, suppose the fruits are ripening, all have ripened, they have been plucked by the farmers and they are in the market now. So there is a tendency that if they are not sold at a particular time, they are not consumed by the public, they will rotten. Okay, so we do not want that. So we simply use gibberellins on the plants so that the ripening period of the plants, the fruits is enhanced. Okay, and the ripening period is enhanced, that is why the shelf life is basically enhanced. So we have, it delays fruit ripening. Okay, so these are some of the functions performed by gibberellins, and the most extensively studied form and one of the first to be discovered was GA3. Alright, I hope this is clear with everyone. Now moving on to the next question. We have the extrinsic factors that play important role in the development of plants are. Okay, so here we have been asked about the extrinsic factors. Okay, pay attention to the term extrinsic. Okay, now what are the extrinsic factors, and before that I would like to discuss what is the role of these particular characteristics, these particular features that have been given, these factors that have been given for the growth of a plant. Okay, so if we take up first of all the nutrients. Okay, we know the nutrients are mainly present in the form macro or micronutrients. Okay, mineral nutrition has been deleted from your syllabus, but you people are aware, you have learnt it in the earlier classes also, that the particular minerals are there. They can be micronutrients, they can macronutrients, okay, that are promoting the growth of the plant, right? So they are very important for the overall development of the plant. So they can be macro or micronutrients, right? And they are mainly present in the soil, from which the roots are absorbing them and they are transporting it to the rest of the plant body, and they are very important for the growth of the plant. Okay, and then comes the plant growth regulators. Yes, the whole chapter is about that. We have so many plant growth regulators. First of all, I will pen them down all for you. Auxin, we have cytokinin, we have ethylene, that is the fruit ripening hormone we were discussing in the earlier class. We have gibberellic acid that we just discussed, and the last one we have abscisic acid. Okay, so these are the plant growth regulators. They can either promote the activity of growth or they can retard the growth activity in a plant, okay, according to the requirements. Okay, next comes the light. Now light is a very important factor. Yes, it is. You know, it is important for photosynthesis. It is required for so many things, right? But the early development of a plant, when it is in the form of a seed, it is below the ground. Okay, there is no requirement of light in the early growth of the plant. Okay, so light is not required for the early growth of the plant until and unless it becomes a small sapling. Okay, before that, obviously it is required after that for the photosynthesis process. Okay, so the only important one that I am writing down, that is photosynthesis, obviously, right? Then comes temperature. Now optimum temperature is very much necessary for the growth of the plants. Okay, if we are taking up the temperature, let's say 28 to 30 degrees. Okay, it is considered as the optimum temperature for most of the plants. Okay, most of the plants very properly proliferate, they grow very properly in this particular optimum temperature. Okay, now what happens if we are saying that the temperature has increased abundantly? Okay, let's say the temperature has striked 55 degrees Celsius. What will be the negative effect of that on the plants? First, I'll write down some of the negative effects over here. If the temperature is high, then what will happen? We can see that the coagulation of protoplasm will take place. Okay, that is the living material that we have, right, for the cell. Then we observe there will be excessive transpiration that will lead to the wilting of the plants. Excessive transpiration will occur. Okay, and we know that there are so many enzymatic activities that are taking place in the plant. Okay, and since they are enzymatic activities, they are very well governed by temperature. Okay, so the enzymatic activities will be affected. Okay, they will be largely affected. So these are some of the negative impacts of very high temperature. So optimum temperature is required. Okay, next comes water. Now what is the role of water for the plant? Water is important for cell elongation. I'll write them down for you. It is important for cell elongation, right? It is important to maintain the turgidity of the cells, okay? It helps to maintain the turgidity of cells, right? And water acts as a medium for so many enzymatic activities, okay? So these are the different factors that are important for the growth of the plant, right? Now, how many of these do you consider as the external factors? Okay, see, nutrients, they are coming from external part only. Obviously, the soil is providing it, right? So the nutrients are external. Plant growth regulators, they are internal. Plant growth regulators are produced by the plant body on its own, okay? They are produced in different parts of the plant. They can be from the roots, they can be from the stem, okay? Then comes the light. Obviously, it's an external factor coming from the sun. Temperature, again the external factor, and water. Obviously, we are watering the plants, okay? Again, it is an external factor. So, the extrinsic factor or the external factor out of these are plant growth regulators, okay? So, they are asking the extrinsic factors, okay? The plant growth regulators are the internal, rest of them are external. So, we have A, C, D and E, and the correct answer for the given question will be your second choice, okay? So, I hope this is clear with you. If it is asked about plant growth regulators, this is the intrinsic factor, okay? This is the intrinsic one. Alright, I hope it is clear. You won't make mistakes again in this. Now, moving on to the next question, that is from Hershey and Chase experiment. During experiment with bacteriophage, Alfred Hershey and Martha Chase used. So basically, what was used in this particular experiment? First of all, you should be knowing that what this experiment was all about. This experiment is very, very significant in the history because it led to the unequivocal proof that DNA is the genetic material. Okay, yes, बहुत सारे experiments हुए थे पहले भी. There were several experiments that were conducted before that, and scientists came to, first of all, they came to this conclusion that the genetic material is not anywhere in the cell, it is in the nucleus, okay? So they narrowed down the location, okay? So we had to find it there. Then there were several, thoughts that can be proteins, because inside the nucleus there is the presence of proteins, there is the presence of RNA as well as DNA, okay? So what is the genetic material, okay? So from that we had to narrow it down whether it is protein, DNA or RNA, okay? So after a lot of experiments, finally Hershey and Chase told from the experimental results that the genetic material is DNA, okay? So what was this experiment? How the particular bacteriophages were used? Let us discuss that, okay? Okay, Makka Priya is saying that it is second, okay? We will come to the conclusion very soon. First of all, you people, yes, you do know the answers, but we are here to discuss the related concept also, okay? So I will discuss this experiment with you. Now, what exactly happened during this experiment? So it is very eminently known as Hershey and Chase experiment, okay? So I will write it down like that only, Hershey and Chase experiment. Basically, in this experiment, they used the T2 phage. Okay, what is T2 phage? It is basically a bacteriophage. And what is bacteriophage? It is a virus that infects bacteria. Right? That infects bacteria. And in this particular experiment, किसकी बलि चढ़ाई गई थी? Which was the scapegoat? E. coli. Okay? E. coli. Now, T2 phage, why they selected this only? Why this particular virus and none other? Because it is having protein coat as well as DNA as the genetic material. Okay? So, it had the presence of protein coat okay, as well as DNA as the genetic material. We had to find whether the genetic material is protein or DNA, okay. So we were simply, so Hershey and Chase were simply conducting this experiment on T2 phage. This was selected because it had both of them, right? Now we know that, okay, I'll display it in front of you. This is the diagram that has been taken from your NCERT. You people are very well about it, very well known about it. Okay, so over here we have been given T2 phages. Okay, this is the situation 1. This is the situation 2. Now what he did? He grew some of the viruses, some of the bacteriophages in the medium containing radioactive sulfur. Okay, so some of them were basically grown in the medium that had radioactive sulfur. So radioactive sulfur is sulfur 35, right? Now the viruses that were grown in radioactive sulfur, see, DNA is not having sulfur. Okay, so will the DNA become radioactive? Obviously not, right? Other medium that was taken was radioactive phosphorus. Now we know that DNA has phosphorus, okay. So in this case, if we are taking up this very medium, will the particular bacteriophage, will the DNA of that bacteriophage will become radioactive? Yes, it will become radioactive because DNA has phosphorus, okay. On the other hand, if we are taking up sulfur, so the protein is having sulfur, okay. So the phages or the viruses that were grown in this particular medium, that is the radioactive sulfur medium, the proteins became radioactive in that particular case. And the other viruses in which the medium or the viruses that were grown in this particular medium in which the radioactive phosphorus was present, in that case DNA become radioactive, okay. So over here we have the first situation, that is for radioactive sulfur, and over here we have radioactive phosphorus, okay. So in this diagram only you can see that the red color is indicating the radioactive part, right? So in this case, red is basically DNA, right? So this is radioactive phosphorus, and over here the protein is red in color, that is radioactive sulfur, okay. Now for this particular experiment, there were 3 steps that were conducted. The very first step was infection itself, okay. So E. coli was infected by this particular phage, that is the T2 phage, okay. So the first step was of infection, okay. In this case, the E. coli is coming in contact with this bacteriophage that will cause the viral infection, okay. Now we had to see that during viral infection, the virus is simply transferring its genetic material into the host, okay. So if the genetic material was protein, the protein would have been transferred in E. coli, and if it was if the genetic material was DNA, it will be the DNA that will be transferred in E. coli, okay. So we saw in the first particular step, we put both of them together, okay. The infection is taking place. The second one is blending, okay. What happens in the blending process? Now the infection has already been caused. The genetic material has been passed to E. coli. Now we have to separate both of them, right? The E. coli has to be separated from the virus, okay. So you can see in this particular second scenario, the virus is getting separated. This virus is getting separated from E. coli, okay. This is the second step, that is blending. And in the third step, what is basically in blending, what they do is agitate it, okay. They agitate the particular setup so that the virus gets separated from bacteria, okay. Now what they will do is the third process of centrifugation, okay. What is centrifugation? At homes, mummy uses cream, okay, to take out butter, okay. So she simply churn the whole cream, and that will lead to the production of, that will lead to the basically coming up of butter, okay. That is churning of the cream. That is centrifugation that is taking place, okay. After centrifugation, what happens? The light weight particles, they come above and the heavy settles down, okay. So the heavier one forms the pellet, Okay, the heavier one forms the pellet and the lighter one forms the supernatant. Okay, the one that is coming up floating, okay, that is coming up and floating, that is the supernatant. Okay, that is forming the supernatant. Now what happened in this case? We observed that the particular bacteria, we are now checking the radioactivity. Okay, whatsoever radioactivity will be found in bacteria, that will particularly show the genetic material, the evidence of genetic material. We observed that the particular bacteria that was infected with the virus that was grown in the radioactive sulfur medium, it showed no radioactivity. Okay, that means this particular bacteria, it is showing no radioactivity. Okay, because the protein part of this virus was radioactive. Was the protein transferring inside the particular body? No, right? That means the transfer was not of the protein, and we know that the genetic material is transferred. That means protein is not the genetic material, right? In the second situation, we observed that the particular viruses that were grown in this particular medium, that is radioactive phosphorus wala medium, we observed that these particular bacteria, they took up the radioactivity, right? They showed radioactivity. So these bacteria were radioactive. Okay, that means they were containing something radioactive inside them. And what was that? It was DNA, right? It never showed radioactivity because sulfur never passed into it, protein never passed into it. That is why we say genetic material is not the particular protein but our DNA. Okay, now they also saw that the bacteria was showing, because bacteria settled down, it formed the pellet part. Okay, bacteria, if we are taking a bacteria, it was in the pellet region. Right, pellet region means the one that is settling down, okay, and the one that is above, that is light in weight, that is above, forms the supernatant. And we saw that these protein shells, that means over here, if we are taking up the proteins, they were present in the supernatant. Because the protein shell detaches, okay, it is not passing into the E. coli. So we saw that in supernatant, the proteins were there. So will the protein be radioactive? Sorry, will the supernatant be radioactive in the first case? Yes, because in this case, the protein was radioactive, it was present in the supernatant, and hence supernatant was radioactive. You can simply see the same in your NCERT also. It has been mentioned, and in the second case it was the bacteria that was radioactive because it simply took up the DNA from bacteriophage. Is it clear with everybody? Now coming back to the question. During experiment with bacteriophage, Alfred Hershey and Martha Chase used what? P32 labeled protein coat and S35 labeled DNA. No, this is opposite. Okay, so it was basically phosphorus, radioactive phosphorus for DNA and radioactive sulfur for protein. Okay, secondly, P32 labeled DNA, yes, and S35 labeled protein coats, yes. So this is the true option. Next is P32 labeled bacteria. It never labeled bacteria, right? It was basically growing the virus in the radioactive apparatus, okay, radioactive medium. So over here S35 labeled bacteriophages, no, so this is also not true. Then comes P32 labeled bacteriophages and S35 labeled bacteria, that is also not true. While we are talking about the labeling, we are simply labeling the ones that we have to detect. What we have to detect? Protein or DNA, so we have to label them. So these are anyway wrong, right? So the correct one is, we had to see DNA and protein. They were labeled with radioactive sulfur and radioactive phosphorus respectively. Okay, I hope this much is clear with everyone. Now moving on to the next question. Next question is assertion and reason. First one, the assertion in this question says mixotrophic nutrition is seen in Euglena, okay, and the reason is Euglena is a photosynthetic as well as chemosynthetic autotroph. Now what is mixotrophic mode of nutrition? That means there is a mixture of something, right? As the name indicates, mixotrophic means when the organism is showing both autotrophic as well as heterotrophic mode of nutrition. Okay, so the mixture of them is called as mixotrophic. Okay, now you can clearly see from this very part only that the reason is not correct. Okay, because कीमोसिंथेटिक ऑटोट्रॉफ्स नहीं होते हैं हमारे पास यूग्लीना की जब हम बात करते हैं. ओके, यूग्लेनॉइड्स आर नॉट कीमोसिंथेटिक, दे आर ईदर फोटोसिंथेटिक और हेटेरोट्रॉफिक. ओके, लेट अस अंडरस्टैंड few characteristic features related to Euglena or simply euglenoids. As I discussed kingdom Protista, I simply separated in different categories, those three particular. The first one was the photosynthetic protists, and under that lies euglenoids. Okay, because they can perform photosynthesis, right? Yes, in some particular cases they are switching from autotrophic to heterotrophic mode of nutrition, but normally they, when in the availability of light, they are performing photosynthesis. Okay, the characteristic features that are very important, that are often asked from euglenoids, I am discussing them with you. First of all, these, since they are belonging to kingdom Protista, they are obviously unicellular. Okay, so here I'll be penning down the characteristic features. They are unicellular, right, and they are having the presence of flagella. Okay, so they are flagellated, that means they are motile. There is one long flagella and one very small flagella. Okay, and next characteristic feature is where they are found. What is the habitat? They are mostly found in fresh water. Mostly found in freshwater habitat. Now, speaking of the mode of nutrition in them, but before that we need to know whether they are having photosynthetic pigments or not. Yes, Euglena has the presence of photosynthetic pigments. I am writing it in brief over here. Photosynthetic pigments. They have the presence of chlorophyll A, chlorophyll B, xanthophylls, as well as carotenoids. Okay, since they are having so many photosynthetic pigments, obviously they are performing autotrophic nutrition. Okay, they are performing photosynthesis. Next is the food that is reserved. Okay, so reserved food is paramylon. Okay, it is important, you should remember this. It is paramylon. Now, another characteristic feature that is very, very important from euglenoids is there is the absence of cell wall. Okay, no cell wall is present. Cell wall is absent in case of euglenoids. Instead of cell wall, there is the presence of another layer named as pellicle. Okay, so if you are asked about the characteristic features, this is one of the most important ones. Okay, the cell wall is absent and in place of that pellicle is present. Now, speaking of the reproduction, sexual reproduction is often not seen, it hasn't been yet discovered in case of euglenoids. So it simply reproduces by reproduction is longitudinal binary fission. Okay, so these are some of the important characteristics that are about euglenoids that you should remember. Okay, and since we are talking about the nutrition in the chapter, in the particular question, so it can be autotrophic because it has the presence of the photosynthetic pigments, but in presence of light. So if we are talking about the situation when the light is present, okay, in the presence of light, The euglenoids will be autotrophic. They will be performing photosynthesis. But in case of the other situation, that is absence of light, euglenoids derive their nutrition from dead and decaying matter. Okay, so they feed on dead and decaying matter, and hence saprophytic. Okay, so the reason statement says Euglena is a photosynthetic as well as a chemosynthetic autotroph. No, it cannot perform chemosynthesis. Okay, so the correct answer for the given question will be your third choice. Assertion is true, but reason is false. Okay, so this is all for today, and the questions from slot 2 next will be carried forward by Mrs. Bushra, and that will be your slot 3, in which she will be discussing the questions from rest of the chapters. Okay, thank you so much, and I'll see you in the next session. Till then, all the very best.